Hey everyone, my name is Michael Muskus and I'm a segment marketing manager here at ATCC. We're so glad you've joined us for the next installment of ATCC's Excellence in Research webinar series entitled ATCC's Best Practices for Optimal Growth and Propagation of Bacteria and Bacteriophages, presented by Jeanette Rimby, lead biologist in bacteriology here at ATCC. In today's presentation, you'll learn about how we here at ATCC cultivate a wide variety of microorganisms and how to achieve ideal growth conditions. A little about our speaker. Ms. Jeanette Rimby has significant experience as a microbiologist with focuses in the fields of bacteriology, genetics, veterinary, and virology. Some of her work has focused on antibiotic-resistant microorganisms and COVID-19 diagnostics. Ms. Rimby is the lead biologist here in bacteriology at ATCC. Her passion for science drives her commitment to advancing microbiology and giving back to the scientific community. During or after the webinar, if you have any questions for our speaker, please use the chat function available through the webinar program. All questions will be answered as allowed at the end of the presentation. And as a reminder, the recorded presentation will be archived on the ATCC website at www.atcc.org. But first, a quick word about ATCC. ATCC is a nonprofit organization founded in 1925. We are headquartered in Manassas, Virginia, with additional facilities in Gaithersburg, Maryland. ATCC is a leading global supplier of highly authenticated biological materials. We're a strong collaborator within the scientific community, and we're an innovator of solutions to enable efficiencies within scientific life science research. So with that, I'd like to welcome Ms. Jeanette Rimby.
Hello, everyone, and welcome to our bacteriology webinar focusing on the fundamentals of bacteria and bacteriophage growth, including how to achieve optimal growth within ATCC's wide variety collection of organisms. I promise to discuss our standards of growth practices within our collection and proper propagation practices in addition to helpful tips and techniques. Furthermore, I will discuss troubleshooting, quality assurance, depositing at ATCC, and available customer resources. But before we start with the fundamentals of achieving optimal growth, allow me to introduce our BioProduction process along with other necessary ATCC manufacturing basics. Here at ATCC, we have over 8,000 microbial type strains, including over 475 reference strains that are recommended for use in quality control. Our collection offers live microbes and derivatives, including inactivated materials and nucleic acids. Specifically, ATCC uses a variety of advanced techniques to characterize and authenticate biomaterials. No single method of identification is sufficient. ATCC organisms are available in 4 different vial formats. First, we have glass ampules, or what we commonly call batch vials. Batch vials hold lyophilized material and are flame sealed with an external glass shell called an ampule that can be stored indefinitely at 4 degrees C. Next, there are serum vials that also hold lyophilized material but have a 5-year expiration and are sealed with a rubber stopper and metal crimp. Finally, our frozen varieties include mini vials that come in packs of 6 vials, and the popular 1 ml frozen cryo vials are both recommended to be stored at -80 degrees C. On the next slide, I will demonstrate how to open a glass ampule. Opening a glass ampule can seem like a daunting task at first. Follow these basic steps, especially paying attention to steps 3 and 4. Score the neck of the ampule with a vial score as seen in the picture to the right. Move the ampule in a circular direction until a fracture line is made. This may take multiple rotations until a fracture line is created. Next, holding the vial upright, begin lightly tapping the outer shell with a metal file along the fracture line. Be sure to collect glass pieces in a sharps container. Relocate product to a biosafety cabinet before removing the cotton plug. Now that we have addressed how to open a batch vial, I will now discuss initiating frozen and lyophilized culture growth. The handling section of our ATCC product sheets provides instructions for rehydrating freeze-dried pellets and thawing frozen vials. When rehydrating pellets, use a single tube with about 5 to 6 mL of broth to rehydrate the entire pellet. Then aseptically transfer the aliquot back into the inoculating broth tube and mix well. When working with frozen material, be sure to thaw vial in a 37°C water bath for about 3 minutes, then transfer the contents to a sterile test tube with the appropriate growth medium. Remember, when working with any live organism, always be sure you are working in a biosafety cabinet after opening the product to avoid contamination issues. In bioproduction, we follow a systematic flow every time a biologist begins a production order. In bacteriology, we work only with BSL-1 and BSL-2 organisms. Step 1, we open the source vial to initiate growth. Then in steps 2 and 3, the biologist makes observations and transfers the item for scaling up depending on the size of the production order. Finally, step 4 is the harvest day, and the product is vialed and sent to preservation. Once the product is preserved, it's QC'd and sent to QA for final approval before shipment. Now that you have seen our different vial types and production process, I would like to showcase what vialing looks like on bioproduction harvest days at ATCC. Production orders range from 20 vials to larger orders of our top sellers being requested in the thousands. Batch vials require a cotton plug to be inserted into each vial manually every time a vial is filled. It's not until the product reaches preservation that the glass shell, called an ampule, is added after lyophilization is complete. Serum vials follow a grid vialing format in which the vials sit in a metal tray before the filling process. Once every vial is filled, the biologist applies a rubber stopper to each vial. Then a porous metal lid and Tyvek covering are applied to the entire tray before sending to preservation. Lastly, frozen vials are filled just as you would guess, unscrewing the cap to add the product and recapping immediately following fill. The vialing process in bioproduction is a very elaborate process requiring advanced techniques and precision. Before I break down bacterial propagation methodologies, I would like to take a moment to discuss atmospheric conditions as a whole and the types of media available depending on the organism. Understanding an organism's atmospheric conditions is very important prior to beginning growth. The typical aerobic organism grows at 37°C or body temperature. Next, microaerophilic organisms require lower levels of oxygen or 5% carbon dioxide atmospheric conditions. Finally, anaerobic atmosphere requires knowing your anaerobes classification, as the presence of oxygen could be deadly for the organism. Also consider establishing a great anaerobic cultivation practice to guarantee optimal growth by using an anaerobic chamber, needle and gas exchange, anaerobic jars, along with anaerobic boxes containing gas packs or sachets. I will break this down further in our anaerobe and microaerophile sections. As we transition into aerobic organisms, I would like to highlight a few of our top aerobic sellers at ATCC and the valuable contributions they provide to science and research. ATCC offers an expanding collection of fully authenticated and characterized food production strains, including a variety of species typically associated with foodborne illness, such as Listeria monocytogenes, Salmonella enterica, and Escherichia coli species, just to name a few. We also offer antimicrobial resistant strains, such as Staphylococcus aureus, for research and drug development. Finally, we offer many multi-drug resistant pathogens, like Pseudomonas aeruginosa. Specifically, we retain cystic fibrosis strains from phase 3 clinical trial studies that provide a unique model system to help understand the molecular pathology associated with cystic fibrosis. These top sellers I just introduced, along with other aerobic organisms, require media with adequate nutrients. Let's now discuss general purpose media for aerobic organisms. At ATCC, our BioProduction teams use simple and nutrient-rich media daily for the majority of aerobic growth. For example, tryptic soy broth and agar is used for the isolation and propagation of a variety of bacterial strains. And the addition of sheep's blood helps with identifying hemolytic activity of the organism. Brain-heart infusion, or BHI, is used for the culturing of fastidious and non-fastidious microorganisms, including streptococci and pneumococci species. And chocolate, or GC medium, is used for growing Neisseria gonorrhea, along with many Haemophilus species. As a reminder, selecting the appropriate media is essential for maintaining the overall health of the organism. Likewise, it's important to know if your organism requires additional supplementation. Here are a few examples of specialized media in the applied organism. Legionella species commonly found in soil and water require a charcoal yeast extract media containing cysteine and iron compounds, which are not commonly found in traditional media. Next, marine media is used for the cultivation of heterotrophic marine bacteria that require unique growth conditions consisting of high salt and low temperature, mimicking a saltwater environment. Azobacter supplement media is enriched in phosphates and magnesium compounds to facilitate growth of these soil-dwelling microbes. Finally, additional supplements to consider are serums from rabbit, horse, and bovine sources, as it is used to support the growth of mycoplasma and ureaplasma. I will break this down further when I discuss mollicutes a little bit later. Now let's discuss anaerobic propagation methodologies and how to achieve optimal growth within agnoxic environments. To grow anaerobically means to grow without the presence of molecular oxygen. In general, anaerobes are classified as facultative, aerotolerant, or strict, with another subset dedicated to methanogens that require nitrogen or ammonia resources. Although many anaerobes are aerotolerant, exposure to oxygen, even for brief periods, can interfere with the culture of some organisms. The next 3 slides are dedicated to strict anaerobes, or obligates, including but not limited to organisms like actinomyces, Bacteroides, Clostridium, and Prevotella species. When growing anaerobic bacteria, the first thing to consider is the nutritional requirements to establish healthy growth. If a self-made media recipe is not giving adequate growth results, the superior choice is to use commercially made, which comes pre-reduced and ready to use. We find that most anaerobic items grow best in broth rather than agar. Specifically, most anaerobes prefer a blood-based media due to its high density of nutrients. Common broth medias to consider are chopped meat, reinforced clostridial, and peptone yeast extract broth with glucose, or PYG. Also consider supplements like 5% sheep, horse, or rabbit blood. Finally, an indicator compound known as rosazurin is added to base media prior to being reduced of oxygen. The media will turn pink at higher redox potentials and will appear clear once the biologist has added appropriate volume of reducing agent. Remember, growing anaerobes can be challenging, but at ATCC, we make use of various technologies for atmosphere control, as well as specifically formulated media. This will be addressed on the next slide. It's highly recommended at ATCC to always reduce your media prior to beginning growth. Here are things to consider when reducing anaerobic media. The typical gas mixture to use when reducing media is 80% nitrogen, 10% carbon dioxide, and 10% hydrogen gas, or what we normally call in bioproduction as 80-10-10. Media should be prepped with reducing agents and stored in an anaerobic environment. Common reducing agents are 3% cysteine, which is generally used for anaerobic cultivation and typically used in bioproduction. Then there is 5% coenzyme M and 15% sodium sulfide, which are frequently used when growing methanogens and some extremophiles. Reducing agents bind to any oxygen in the media, which prevents the oxygen from interacting with anaerobic organisms. Drawing your attention to the first picture to the left, Hungate tubes are typically used when gassing out test tubes. They have a screw cap and a butyl rubber stopper that allows gas exchange through a needle and syringe. When reducing media in bottles, add the appropriate reducing amount, as seen in picture 2, and then relocate bottles and agar plates to an anaerobic jar and connect the jar to a gas generating system or a noxamat. That will rapidly remove oxygen and replace it with the precise anaerobic gas mixture as seen in picture 3. Two final recommendations are to pre-reduce media 24 hours prior to inoculation to allow acclimation of reducing agent within media. Lastly, pre-reduced media and organism should be open in an anaerobic chamber to ensure a constant flow of oxygen-free gas as seen lastly in picture 4. For further detailed information, please check out my 2024 Best Practices Anaerobic blog entitled Overcoming the Challenges of Growing Anaerobic Bacteria. And keep in mind that ATCC has over 1,000 active anaerobic holdings, including over 400 type strains. And within our collection, we even have strains isolated as part of the Human Microbiome Project. Next are organisms that can thrive in hostile environments, known as extremophiles. These types of microorganisms are environmental anaerobes, which grow in niche environments that are acidic, excessively hot, or irregular in air pressure. Organisms, like thermophiles, are found in geothermic heated regions, like hot springs in Yellowstone National Park, or high salted regions, like the Great Salt Lakes or the Dead Sea, known as halophiles, pictured on the Petri plate. Growing these kind of organisms may require Bouch or SRM bottles. The bottle is a serum bottle with a crimped aluminum seal that helps maintain additional pressure for culturing. Also note that when growing extremophiles, the gas mixture requirement may also need to be free of carbon dioxide, which is different from traditional anaerobes as previously discussed. Please note if you have further growth questions, feel free to reach out to our customer service team. Quite the opposite of extremophiles are organisms known as microaerophiles. Microaerophilic bacteria require lower levels of oxygen than those found in standard air, with typical oxygen levels between 5 to 10%. These organisms need oxygen because they cannot ferment or respire anaerobically. However, they will tolerate high concentrations of oxygen. Some common microaerophilics include helicobacters, which prefer biphasic growth. To grow biphasically means the organism requires a solid surface to adhere to, but grows primarily in the liquid-to-air interface. The medium consists of both a solid and liquid state in a single culture tube or tissue culture flask. Next, Neisseria, or fastidious organisms that grow best on chocolate agar with an atmosphere at 5% carbon dioxide. To grow Campylobacter organisms, many species require Brucella broth or TSA medium, which has been supplemented with formate and fumarate. It is also beneficial to use a reliable gas generating system to create and monitor the desired levels of oxygen and carbon dioxide. Lastly, here are two final recommendations to consider. Some microaerophilic species may require incubating in anaerobic boxes with gas pack sachets. Also make sure the media is pre-warmed before inoculation to avoid growth issues. And finally, Did you know microaerophiles were traditionally cultivated in candle jars? Essentially, a lit candle was introduced before sealing the container's airtight lid. The candle's flame burns until extinguished by oxygen deprivation, creating a carbon dioxide-rich environment. To say the least, you now know the old school and the new school methodology of cultivating microaerophiles. Now, switching gears a bit, I would like to discuss the natural enemy of all bacteria, the phage. Bacteriophages are bacteria eaters, and more exclusively are host-specific viruses that infect bacteria cells. In bioproduction, we use broth propagation methods routinely, but Adam's agar overlay method is acceptable as well. Propagations take place over 4 days. Starting on day 1 by propagating host using basic cultivation practices. On day 2, start by amplifying the host and phage. Ensure the host is in early log phase before adding lysate. Then on day 3, vacuum filter the lysate culture to rid of bacteria cells, followed by performing a spot titer to determine the plaque-forming units, or PFU. Finally, day 4, the titer plates are read. At the higher dilutions, individual plaques should be countable to determine adequate titer. On the next slide, I will show what a titer plate looks like following a serial dilution. But before we transition, consider these quick tips when propagating phage in the event you don't see plaques. First, confirm the host is in early log phase prior to introducing the lysate. This can be checked by using a spectrophotometer. The optical density should read between 0.1 to 0.3 OD. Determining the appropriate multiplicity of infection, referred to as MOI, may also be necessary to optimize phage infectivity. The MOI tells the ratio of the number of virus particles to the number of target cells present in a cell culture at the time of infection. Additionally, try adding 2+ salts like magnesium chloride and calcium chloride to the media, which increases the potential for phage aggregation through ionic bonding. But above all else, remember starting with a healthy host is essential for phage propagation. After performing a spot titer, this is what a typical titer plate will show the next day: plaques. Plaques are the zone of clearing seen within the lawn of bacterial growth following standard spot titer methodology. Here we have examples of spot titers after a serial dilution is performed. All plates were incubated overnight and read the next day. Within the zone of clearance, some resistant colonies may also be seen, which is normal. Each plaque picture has distinctive plaque characteristics and PFU concentrations. Depending on the phage's life cycle, that can have a great effect on the plaque growth, as well as other physical and chemical characteristics. The final propagation methodology I will discuss will be the cultivation of mollicutes, which in some cases may be the most challenging of growers. They are a class of bacteria that are distinguished by their lack of a cell wall. Rather, most members of this class contain sterols in their cell membrane to increase membrane rigidity. Most are aerobic or facultative anaerobes. Mollicutes tend to require a rich growth medium and are often sensitive to overgrowth. And most species may not grow on agar, but growth in broth is rarely more than lightly turbid. Broth media is highly enriched and includes a pH indicator to track growth cycle. Always consider the pH of the media when working with mollicutes. Growth time can take weeks with some species. It is often favorable to inoculate media with a supplement of fetal bovine or horse serum to help maintain cell membrane integrity. Also, it is recommended to use at least a 10% inoculum when scaling up growth with mollicutes. If an organism is struggling to grow, try adding 10% fetal bovine serum as a supplement. Specifically, with organisms that lack a cell wall, the serum can function as a pH buffer and can inactivate proteolytic enzymes. As we wrap up the fundamentals of achieving optimal growth in bacteriology, I would like to take this time to spotlight our manufacturing science and technology team, known as MSAT BACT. They perform many tasks to ensure our organisms are of top-notch quality and are the experts to consult when troubleshooting. Their methodologies include growth optimization assays and comparing preservation methodologies to determine the best growth practices for numerous organisms. MSAT also conducts special troubleshooting methods, including antibiotic resistant testing or adapting new PCR protocols to test for the presence or absence of antibiotic resistant genes, toxin genes, or any gene of interest for characterization. Lastly, new accessions first pass through the team to determine ideal growth conditions for new deposits based on depositor notes and research. As part of our commitment to provide well-characterized and authentic organisms, ATCC is completing whole genome sequencing on all new accessions. The complete and annotated genomes are then made available on our ATCC genome portal. Finally, I would like to highlight our microbiology quality control testing team. They use a variety of advanced techniques to characterize and authenticate our BioProduction organisms. Tests include phenotypic analysis through visual observation, and proteomic analysis is tested using multi-TOF mass spec pictured to the left. Functional analysis testing is used for the detection of drug-resistant and virulent genes. Finally, there is also genotypic analysis through sequencing conserved regions of the genome and whole genome sequencing. It is through the utilization of these standards that researchers look at ATCC for a wide range of authentication resources to safeguard reproducibility and to meet the requirements for funding, publication, and quality control. In 2019, ATCC launched its Genome Portal, a cloud-based platform that allows users to browse our authenticated and traceable reference genomes. The ATCC Genome Portal is the only authenticated reference genome database for ATCC microbes. As of today, we have completed whole genome sequencing on over 5,000 microbial items and published the reference genomes on our Genome Portal. Customers who purchase our strains will have access to the corresponding reference genome. We also offer a supporting membership for customers who are interested in accessing and downloading our entire collection of genomes. And if you are wondering how to make a deposit at ATCC, please follow these easy steps. First, visit our company website, atcc.org. Then choose the type of deposit you are wishing to make. Next, fill out the electronic deposit request form provided. Finally, our content and accessioning team will review the submission, and someone from the team will reach out with further details upon deposit being approved. At ATCC, we offer 4 deposit service options. The first type is a general collection deposit, which is a great way to preserve your research biomaterials and make them available for worldwide research. Next, we also offer National Park Service and patent deposits. Lastly, type strain deposits, in which after ATCC conducts viability and identity testing, the depositor is provided with a certificate of deposit for publication purposes. Upon receiving ATCC product, consult our website to access the item's product sheet. First, search for your item using the search bar at the top of the page. From there, you can locate the product sheet PDF. The reference material includes helpful instruction pertaining to maintenance media, atmospheric needs, incubation time, and other growth guidelines. Here are additional resources to use when beginning microbial propagation and cultivation. You can access culture guides from the following sites: atcc.org/guides and atcc.org/webinars for bacteriology and introduction to microbiology resources. Also, please check out our blog posts with topics concerning best practices, environment, global health, and research highlights. Additionally, please visit our support page for frequently asked questions and direct contact services. In conclusion, I have discussed our bioproduction process as well as breaking down the fundamentals of achieving optimal growth with aerobic, anaerobic extremophiles, microaerophilics, bacteriophage, and mollicutes. Now I would like to review a few best practice recommendations. First, to always follow a standard propagation method and utilize the ATCC product sheets as a guide for helpful handling information. Next, when growing any organism, be sure to use appropriate medium, growth conditions, and length of incubation. Also be aware that certain organisms may prefer shaking incubation over traditional static incubation. Pay attention to commercial versus self-made media, as commercially made media will often help with poor growth. Don't forget to reduce anaerobic media prior to inoculation. Always take the pH of the media into account when working with mollicutes, as their cell membranes rely on it. When growing microaerophilics, it may be beneficial to use a reliable gas generating system. Finally, if you don't see plaques while propagating phage, ensure the host is viable and consider adding extra salts to the growth medium. Thank you for attending this presentation. The bacteriology team shows their commitment to maintaining ATCC's continued demand for high level of expertise, methods, standards, and services, branding us as the largest global biological resource center. A big thank you to the following people and teams. Graham Hogue for his dedication and technological savvy contributions to this presentation. To Team Bacteriology for helping capture the laboratory photos used throughout this webinar presentation and for sharing their expertise in microorganism growth. To Nancy Kruger, who set the bar high with her broadcasts of Bacteriology, the Fundamentals in 2020. To Joe Theriot for his mentorship and quick rallying to dedicate time to our webinar. And finally, a round of applause to our microbiology bioproduction teams whose commitment and hard work allow ATCC to support innovation in the scientific community. The teams include bacteriology, mycology, protostology, molecular genomics, and MSAT-BACT.
Thank you, Jeanette, for the excellent presentation. In a few moments, everyone will begin our Q&A session. To ask questions, please use the chat function available in the webinar program. In addition to Jeanette answering questions, we also have with us Joe Theriot, Lab Operations Manager, and Graham Hogue, Associate Biologist from the Bacteriology team. We also have with us a Program Manager from the Genome Portal. And remember, the webinar presentation will be available on demand at www.atcc.org.
First question that we have that came in is probably for Jeanette and the bacteriology team. Why do you need to infect bacteria with bacteriophage at early log phase and not mid or late log phase?
So great question, and this often comes up too for those that have been doing phage work for a while or maybe have just decided to start beginning. It's important that the host be at that optimal early growth phase. It just increase the infectability of the bacteriophage as a whole so that you can get a great concentration in the end. So essentially, if your host is entering that late log phase into the stationary phase, it's going to be a lot harder for the host, for the phage to actually propagate and keep reproducing. So essentially to keep infecting.
Great, thanks, Jeanette. The next question, what are your recommended protocols for detecting and eliminating contamination in bacterial cultures or phage stocks?
Yes, another great question. Again, make sure that you are working in a biosafety cabinet whenever you are working with any live organism. It's essential. Also, make sure that your media has been autoclaved as well before beginning any type of inoculation with a live organism. So oftentimes, media that maybe has set out at room temperature for too long too, it's lost its nutrient value, and the organism can no longer thrive within its environment.
Great, thank you. So here's another question that came in. What's the most important steps to consider when growing anaerobic bacteria?
Yeah, definitely. Make sure that you know the type of environment that your anaerobe is essentially accustomed to living in. So if it is a strict anaerobe, then that means that it cannot be in an oxygen or an oxygenated environment. So make sure that you are aware of that, and to also remember to reduce your media. So regarding that, you can always find that kind of information on our product sheet within our company website, and we outline exactly what type of media to use, as well as the reducing agents and the specific gas that is required for that organism to thrive.
Great, thank you. Let's see, here's another question. Okay, this is probably a little bit general, but why doesn't an organism grow the same way every time?
It's not a general question, and in fact, it's a question I think that's often, brought up in any kind of microbial conversation. So what I always resort to is, as I know you're probably aware, bacteria undergo cell division. So the growth rate of the bacterium is measured by determining the change in bacterial number per unit time, or what is called generation time. So E. coli, for example, has a generation time of about 20 minutes, versus Mycobacterium tuberculosis that has a longer generation time of 20 hours. So the growth rate can be affected by many factors, and this can include pH, temperature, available nutrients, atmospheric conditions, light as an energy source, so whether it's phototrophic versus chemotrophic, and even osmotic pressure, and of course the characteristic of the organism. Also consider mutations and genetic drift. So this can greatly affect over time what can cause gene variants to disappear altogether.
Great, Jeanette, thank you. We are getting a lot of questions coming in, so
Perfect.
yes. So you and your team are going to be on the call for a while, so this is great. Another question we have is, do you have troubleshooting tips for poor organism growth and recovery?
Yes. So check your culture conditions and identify any differences or problems with culture technique, incubation, and media. Always check your protocol and confirm all steps were followed, including proper storage, thawing, and freezing of cell products. Cell cultures fail for one of two reasons, essentially: culture conditions or the cells themselves. So microbial contamination is a major cause of cell culture failure and may take some detective work to determine root cause of also the contaminated source.
Great, thank you. Let's see, how about, it says, I would like to know how to preserve anaerobic cultures after the second passage.
Okay, so obviously the organism has already been opened, and assuming that you're using a good anaerobic cultivation practice, so whether the anaerobe is being kept in a glass jar or a cultivation jar, so to speak, to ensure that all the adequate gas mixture is still within the environment of that organism. Make sure, too, that if this is a strict anaerobe, you're working in a anaerobic chamber at all times when handling a strict anaerobe to ensure that you're getting that constant flow of oxygen-free gas. It is essential. And again, knowing once again the classification of the anaerobe. So if it is facultative, sometimes they can tolerate oxygen for briefer periods of time than obviously a strict anaerobe.
Great, thank you. Here's one. This is more for the genome portal. So I know you showed a couple slides for that. And I believe we have Brianna from the genome portal team on the call. So Brianna, we have a question that came in. Is ATCC doing whole genome sequencing on all the microbes in the collection?
Yes, that is our long-term goal. So to date, we have 5,000 items published to the ATCC genome portal. And here in a few weeks, we're going to publish another 250 genomes.
Great, great. Thank you. Thank you, Brianna. Okay, we'll go back to Jeanette and team. What is the best way to collect an extremophile sample and preserve it prior to culture?
All right, so with extremophiles, you really have to research the history of the organism to make sure that you understand exactly what type of an environment that it is accustomed to, whether it needs to be a certain aliquot of carbon dioxide, so whether it's 5% CO2. That's something to always consider. And I also mentioned during that talk too about SRM bottles or Bouch bottles. That can also help with just with growth in general for the extremophile to ensure that if oxygen is perhaps a limiting factor for its growth, then that can help essentially give it that extra protection during incubation.
Great, thank you. Let's see. Okay, here's one. It says, recently I'm working on E. coli and MS2. They worked for me for a while, but I'm having trouble now. Can you please share some tips on troubleshooting? I think you've kind of covered this before, but if there's anything in specific regarding E. coli and MS2, Jeanette, that might be a little too specific. So if it is, we can always follow up with the questionnaire.
Yeah, so the main thing with E. coli is that its generation time is very rapid. So you want to make sure that the cells are in fact viable depending on how long you're incubating. If it's longer than 24 hours, you might want to scale back and start checking the cells perhaps at an earlier time slot, maybe at the 12-hour mark. So that can easily be done using potentially a plate reader or some type of spectrophotometer that just check exactly what is the optical density of those cells.
Great, thank you. What are your recommended protocols for detecting and eliminating contamination in bacteria cultures or phage stock?
Yeah, so essentially whenever you're working with phage, once you have actually been able to amplify and propagate the phage lysate culture, you want to make sure you're filtering the next day to rid of any type of additional cells that are within the culture itself and to get rid of any other perhaps spores that may be occurring within the vicinity of that culture. So I usually promote vacuum filtering. It's very fast, it's easy generally whenever you are growing bacteriophage. So look into vacuum filtering, and the pore sizes generally you start with are a 0.45 micron filter and then move down to a 0.2 micron filter to just ensure that that lysate culture is rid of any type of other cells.
Great, thank you. Let's see, another one came in. I believe this one's probably for Brianna. How do I find the genome for a strain that I purchase?
Yes, you can go to genomes.atcc.org and you can search by catalog number. Additionally, you can go to the product page, and if we have sequenced the genome, there'll be a link out directly on the product page that you can just click on that, and then it'll bring you to the product page on the genome portal. And then you would be prompted to log in and then type in the lot number for the item that you purchased. And once you do that, then the genome will download for you.
Great, thank you. Jeanette, back to you and your team. What is the most challenging bacteria that you grow?
I would probably say mollicutes would be very challenging, just because you have to be aware of they lack a cell wall. So essentially, an organism that doesn't have a cell wall, you have to also think of, okay, then that means more or a harmful type of contaminants could potentially leak in. So again, using great aseptic technique always and working with mollicutes is essential. Maintaining the pH of the media itself and working with mollicutes, always keep that in mind.
Great, thank you. The next question we have, does your team grow all the bacteria in your inventory?
Yes, so we actually do, as far as the business needs are concerned.
All right. Next question. What's the appropriate temperature to store your organism?
So again, as I stated earlier, making sure that you know what essentially type of vial type that you have. So whether it's a cryopreserved organism, so that would be considered frozen. And we recommend them frozen vials, cryo vials, to be stored at minus 80 degrees C. The batch vials or serum vials, perceptorol vials, they can be stored at a 4 degree C temperature. But again, whenever you're working with your organism, make sure that you are reading the product sheet and just making sure you are aware of what the correct information that is given on how to store your microorganism, because we do state that on our product sheet.
Great, thank you, Jeanette. And this might be in line with my next question, but since it's been submitted, I will ask it. But propagating frozen bacteria, yeah, propagating frozen bacteria from ATCC is become more difficult. Very small colonies. Any suggestions?
So yeah, I would definitely make sure that you are using the appropriate medium first, and that the media has not passed expiration as well. Whenever you're thawing your vial, make sure that you're using a 37-degree water bath, and make sure that the vial isn't sitting out on your bench for far too long of a time. So once you know that you're ready to work with the organism, make sure that you have everything set up within your BSC to automatically move from one phase to the other, so you can get the organism into the appropriate medium and start the incubation process.
Great. Thank you, Jeanette. Had a few questions come in about sharing the slides for this webinar, and we will be sharing those, and I'll let you know where you can find those once we wrap up the Q&A session here in a few minutes. Next question would be for Brianna, for the Genome Portal. How close of a match do you require to pass QC testing with whole genome sequencing? Are some bacteria more difficult to extract high-quality DNA for sequencing?
Okay, so regarding the extraction, so yes, we use different extraction methods for different organisms. Some organisms are quite easy to extract. Other ones, it will take a much higher count to actually get enough DNA for our sequencing process. For example, our bacteria, we actually run on 2 different platforms. We run all of our bacterial samples on both Oxford nanopore sequencing to get the ultra-long reads, plus we run them on Illumina, and we use the Illumina and the ONT sequencing to produce a hybrid assembly. So for ONT, we need high-quality extraction. So again, depending on the organism will depend on how we extract it. Regarding the first question was, yes, the QC match. For some of these organisms, there is no actual public database that has them. Some of our type strain deposits, we are doing sequencing on them for the very first time. So that is the baseline. That is your reference sequence. For other items, they've never been sequenced before in our collection. Again, we're almost 100 years old, so we have items that we're sequencing, and we're discovering that they may need to go through a name change. So we'll do sequencing on them, and they may sit, and we have to go through a process where it goes through name change just to update the taxonomy. I hope that answers your question.
I think it does. Great. Thank you, Brianna. The next question we have is, what is the favorable temperature for bacteriophage to propagate?
So typically when working with phage, we're looking at around, and again, 37 degrees C is typical if you're working like with E. coli phage, for example. But some Pseudomonas phages, they prefer more of that environmental temperature of 27 degrees C or 26 degrees C. So knowing, again, the actual environment that the host thrives in is the first step. And to identify that can always be found on our product sheet. We state that whenever we first begin with propagation of the phage, and that is to first establish a good, healthy host.
Great, thank you. Here's another question for you and your team, Jeanette. Why does mixed culture bacteria often look so different when isolated out, even under the same environmental conditions?
Yeah, it really is kind of a natural phenomenon that can happen. So bacteria are just predisposed for millennia to adapt and overcome to any type of circumstance. So if the culture is mixed, then you have 2 different types of competition that could be happening. And it also just depends on what the culture is mixed with, whether it's E. coli versus Salmonella. It depends, again, on the microorganisms. So, but once you plate them, again, the competition for the resources. So whoever can outcompete the other using whatever type of signaling system that they are trying to essentially increase their longevity. So only the fittest microorganism will win in the end, but during that process, you can always see the different types of chemical, metabolic reactions that could be happening. So, and you can see that in the colony types, colony size, the actual tint and color of the colony. So, yes, and isolating those colonies too, just to figure out exactly what type of organism it is, and that can be done easily just doing a 16S sequencing.
All right, great. Thank you so much, Jeanette. And with that, I think we're going to wrap it up. That is a lot of questions. We had about 20 come through. So great engagement, everybody. Thank you so much for joining the webinar and the Q&A session. Jeanette, I'd like to thank you and your team and Brianna for answering questions from our audience.
Yes, thank you so much. This was quite the honor and privilege to be able to do this.
And everyone, you can find the slides and this webinar on demand available on our website early next week. That would be www.atcc.org/resources. I'll also say, please be sure to sign up for our monthly newsletter. and plan to attend our next webinar. We usually have at least 2 every month. So again, thank you all for attending today's webinar, and have an amazing day.